class, and we are continuing with photosynthesis. So let us continue with the light reaction of photosynthesis. So what happens which happens in the grana of the chloroplastid as well as the fret or the thylakoid or the fret or the stroma laminalis of the chloroplastid. So let us discuss about it. We see here, this is the grana, these are the thylakoids, clear, and these are the stroma lamellae, which are the region where the light reaction occurs. So if we go on for the location or the region where light reaction occurs, it is the grana and the stroma lamellae. Now in light reaction there are two types. One is the cyclic photophosphorylation and other is the non-cyclic photophosphorylation. The non-cyclic photophosphorylation occurs in the appraised grana. Appraised grana means the grana which are present in between the upper and lower thylakoids. These thylakoids which are present in between upper and lower thylakoid, these are called appraised thylakoids of the grana. So this is the region where the non-cyclic photophosphorylation occurs. But cyclic photophosphorylation occurs in the unappraised thylakoid, that is the upper thylakoid, lower this one, the stroma lamellae. These are the region where the cyclic photophosphorylation occurs. So this is the location where we find cyclic and non-cyclic photophosphorylation. Now we will discuss what is cyclic and non-cyclic photophosphorylation. So first of all, let us discuss about the non-cyclic photophosphorylation. You can see here non-cyclic photophosphorylation, the scheme appears to be in Z-like, so it is also called Z scheme. Now what is photophosphorylation? The formation of ATP. This formation of ATP from ADP and inorganic phosphate is phosphorylation. Clear? And when the required energy for this formation of ATP, because we know that adenosine triphosphate is a highly energy rich molecule, because this bond when ADP combines with inorganic phosphate, it forms the adenosine triphosphate, and this particular bond stores a lots of energy. So it is an energy rich compound, and that energy for this formation of this bond comes from the light. Clear? So it is called photophosphorylation. Now here none of the electron fulfill its own hole. Clear? So that means there is no cyclic movement of the electrons. Never the electrons come back and fulfill its own deficit. So it is called non-cyclic photophosphorylation. Now we will discuss describe the non-cyclic photophosphorylation. Now first thing is that non-cyclic photophosphorylation involves two photosystems, that is photosystem 2 and photosystem 1. So this is photosystem 2 and this is photosystem 1. You can see here. This photosystem 2, we know that it is 680. P680 is at the center, which is surrounded by 200 to 300 accessory pigments. Clear? Now light falls on these pigments. Actually basically both the photosystem is active at the same time, but for our convenience of a study, we are studying it one after another. So light falls on this photosystem PS2, and the accessory pigments absorb various wavelengths of the light and finally transmits this light energy to chlorophyll A. Now the chlorophyll A, so accessory pigments absorb various wavelengths of light and transfer it to chlorophyll A. Chlorophyll A, the atoms of chlorophyll A, they have the electrons in their outer shell, and these electrons are present in a particular orbit and is considered to be in the ground state. Now this orbit is nothing but an energy level. They are present in that particular orbit because they have that particular energy level. clear, and that is said to be in the ground state. Now when this huge amount of energy passes to chlorophyll A, these electrons which are present in the atoms of this chlorophyll A, in the outer shell of the atoms of this chlorophyll A, they absorb this huge amount of energy and they get excited. So from ground state, now they are in excited state. So these electrons which were in ground state, after absorbing the light energy, they are in excited state, and as a result they are not fit for that particular orbit. So they are deflected from that particular orbit, and when they are deflected from their particular orbit, they move to some primary acceptors. So this is the primary acceptors where the electrons move first, and therefrom it moves along the electron chain, transport chain, clear, where there is cytochrome B3, cytochrome F, and various electron acceptors. Now when it moves down the hill along the electron transport chain, what happens? This excess energy which it has gained and has got deflected from, so that energy it is releasing, and that energy which is released by these electrons is utilized by ADP and inorganic phosphate to form adenosine triphosphate. So that energy which is released by these electrons are used by ADP and inorganic phosphate to form ATP, adenosine triphosphate, clear. And now when it is releasing its energy to form adenosine triphosphate, so this is phosphorylation, and since the energy the electron has gained from light, so this energy is providing ADP to form ATP, so it is called photophosphorylation. Now this electron, after releasing the excess energy, now it has returned to its ground state from excited state. Now here also simultaneously light is falling, clear. In PS1 light is falling, the accessory pigments are absorbing the light and it is transporting it to chlorophyll A, P700, and the electrons here also from get deflected. So as a result, a gap or deficit of electron has been created here in chlorophyll A. These electron which has already returned to its ground state now fulfill this deficit, clear, and thus the electron gap, now it is fulfilling this electron gap or electron deficit. The electron which has been deflected from this PS1, clear, or photosystem 1, P700, chlorophyll P700, it is moving to again a primary acceptor, and then NADP, that is nicotinamide adenine dinucleotide phosphate. So it is moving there and it is combining with it, forming, and proton, this NADP is now it's added with a proton and form NADPH, which is a reducing agent, which will act as a reducing agent and is stored for the future use. Now next is another thing happens in the excited chlorophyll in PS2, it splits water. This is called photolysis of water. It splits water molecule into proton and oxygen, that is hydrogen ion and oxygen. Oxygen releases its electrons, and that electron which is released from oxygen, that is fulfilling the electron deficit which has been created in photosystem 1, clear, and this oxygen is finally evolved. So the oxygen which is evolved during photosynthesis is coming from water only, not from the carbon dioxide. And finally, so, and these protons are, it can neutralize NADP to NADPH2, which can act as a reducing agent. So this is, and metabolic water is also formed, because this is actually which is forming hydroxyl ion. Hydroxyl ion is releasing this electron, forming hydroxyl radical. Hydroxyl radical combines, 4 hydroxyl radical can combine to form 2 molecules of water and 1 molecule of oxygen, which is released, and water forms the metabolic water. So this is about the non-cyclic photophosphorylation. It is also called Z scheme. It is, you can see here from the electron is moving, and it is fulfilling the deficit in photosystem 1. From photosystem 1, the electron is moving to NADPH, and from water, by the splitting of water, electron is moving to photosystem 2. So none of the electron is returning to its own place. So this is called non-cyclic photophosphorylation. Another photophosphorylation occurs, already we have discussed, it occurs in the stroma lamellae and unappressed thylakoids of grana, where only photosystem 1 is involved. Only PS1 is involved, you can see. The accessory pigments here also absorb light of various wavelengths, passes it to chlorophyll A, P700. Clear? The electrons in similar way here also gets reflected, and the deflected electrons move to primary acceptor, and then through a series of electron acceptors it moves, and during this movement it releases its excess energy. That means the energy which has made it excited, it is releasing that excess energy, which is utilized by ADP and inorganic phosphate to form ATP. Clear? And now it is again returning back to ground state, and it is fulfilling the deficit which has been created in here. So here the electron which is moving out is again coming back to fulfill its deficit. So it is cyclic photophosphorylation, where only ATP is produced and NADP is not produced, water is not splitted up. Clear? And it occurs in the unappressed grana and the thylakoids. So here we can see the difference in non-cyclic photophosphorylation. The photosystem 1 and 2 both are involved, but in cyclic photophosphorylation only photosystem 1 is involved. Next is the electrons that are expelled from, in non-cyclic photophosphorylation, the electron that are expelled never returns to its own position, but in cyclic photophosphorylation the electrons which are expelled again return back to its own position. In non-cyclic photophosphorylation the products are ATP and NADPH2. ATP is the energy source and NADPH2 is the reducing agent, but in cyclic photophosphorylation it is only ATP. The non-cyclic photophosphorylation occurs in the appressed thylakoid of the grana. Clear? But cyclic photophosphorylation occurs in the unappressed thylakoids and stroma lamellae. Unappressed thylakoids of the grana and the stroma lamellae. In non-cyclic photophosphorylation there is liberation of oxygen. Clear? O2 is liberated. Here O2 is not liberated. Water is not there is no photolysis. In non-cyclic there is photolysis of water with the liberation of oxygen, but in cyclic photophosphorylation there is no photolysis of water with the liberation of oxygen. So these are the differences between cyclic and non-cyclic photophosphorylation.